This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 76 for April 2nd, 2010. Hey everybody, I'm Vincent Racaniello, and this is TWiV. Today I'm speaking with one of the world's experts on retroviruses, Dr. Stephen Goff, professor of microbiology and immunology and biochemistry and biophysics here at Columbia University Medical Center. He's also an investigator of the Howard Hughes Medical Institute. Dr. Goff's laboratory is interested in the replication of retroviruses, in particular the cellular gene products that are required for their replication. He obtained his PhD from Stanford University, where he worked in the laboratory of Paul Berg, and there he participated in the first recombinant DNA experiments. Subsequently, he did postdoctoral work with Dr. David Baltimore at MIT, where he began to work on retroviruses. I met Steve in 1979 when I joined the Baltimore laboratory. Steve came to Columbia in 1981, and I followed him here the next year. Our laboratories occupy the same floor, and his office is adjacent to mine, so I can always pop in to talk about viruses. Dr. Goff recently turned his attention to Xmrv, the newly discovered retrovirus that has been implicated in prostate cancer and in chronic fatigue syndrome. He recently published the observation that Xmrv does not replicate well in a variety of cell lines, but infects LN-cap prostate tumor cells very effectively. This robust replication may be a consequence of enhanced production of viral mRNAs in these cells. With the increased focus on Xmrv, I thought it would be a great time to hear Dr. Goff's thoughts on the virus and its involvement in human disease. Stay tuned after our conversation for some closing remarks. This episode of TWiV is brought to you by Data Robotics, the makers of Drobo. The Drobo family consists of 4 different models for data storage. Each one is like a big hard drive that never gets full and never loses data. You can start with 2 drives and add more, up to 4, 5, or even 8, depending on the model. Now, I have 3 Drobo units, as you've probably heard. 2 of them are home to protect my collection of family photos, TWiV and TWIP episodes, videos, and writings. At my lab, I keep all my data on a Drobo. When I copy a file to a Drobo, I know it's safe because if any drive fails, I simply pull it out and replace it with a new one, and the data are automatically restored. There are 4 Drobo models. The Drobo model has 4 drive bays. It's great for backing up your iTunes library, photographs, or videos. I have a Drobo in my lab that I use to back up crucial data that I can't lose. My data is my career. I can't afford to lose it, so I put it on a Drobo. You can save $75 to $500 on the purchase of a Drobo, Drobo S, Drobo Pro, or Drobo Elite at drobo.com/TWiV by using the coupon code TWIVPOD, all in capital letters. Or you can try to win one. We're fortunate that Data Robotics has given us 3 Drobo S units to give away. We've finished the second giveaway, and we're in the process of marking the entries. And we're about to begin the third. Go to twiv.tv/drobo-contest for details. Now, on to my conversation with Dr. Stephen Goff. I wanted to talk about Xmrv with you.
Okay.
We could probably talk about all we know in an hour, right?
Yeah, it's probably true.
It's not a lot of papers on PubMed, but it's a new field. Why don't you tell us where this virus originally came from? Was it picked up in a prostate tumor, right?
Right. So, the ultimate origin of the virus is a topic of discussion still, but the history of its discovery is, as you say, that it was found in prostate cancers. It was one of the first new viruses found in humans using virochips, where people were looking for new pathogens. and Ganem and DeRisi and the people at UCSF with Bob Silverman were looking especially in prostate cancer because of a genetic history actually in humans. So humans have a familial tendency for prostate cancer that they tracked to a gene in innate immunity. And so there was the plausibility that this disease had a component that could be infectious.
So that's why they were looking for that.
So that's why they
I
looked.
see.
Now that hasn't held up well, actually, anymore as the basis for the disease, but it was the reason they looked, and coincidental or lucky or not, they found this virus.
So this was the only virus they found in prostate tumors?
I don't know. This was the strikingly correlated one. That this, in their initial look, was present in a fair number of the familial prostate cancer people, and not in most of the controls. So this was the virus that correlated most strongly with the disease.
So I remember when this was picked up, and you began to work with it in your lab shortly afterwards.
Right.
Which I know because you're right across the hall from me. And I don't remember anyone discussing its origin at that time. That seemed to have come much later when chronic fatigue entered the picture. But when it was first picked up by DeRisi and Ganem, did they realize that it was probably a mouse of mouse origin?
Yeah, I mean, that was certainly the thought. I don't think it was the first topic people addressed, but the sequence similarity is so great to the mouse xenoviruses that I think everybody assumed it was going to have arisen from a mouse.
So it's about 30 bases different from a mouse virus?
Yeah, it's incredibly close now. I forget the number, 98-point-something% identical to the mouse xenoviruses.
Could it have been a contaminant?
So that was also a concern, and it still is in a lot of labs. The levels are low. People are always using PCR to detect it. And when you're in a lab that's working with large amounts of any virus, PCR contamination is always a problem. So people do have to be very careful, even paranoid, to make sure they're avoiding it. But I think at this point it's not likely to be a contaminant in most of the assays that we're seeing, because for one thing, it's not identical. So John Coffin and crowd have looked in a very large number of mouse species.
Lab species and wild, or just lab?
Both, actually, mostly lab.
Okay.
But all mice have about 10 or 20 copies of the xenoviruses in their genomes. And all the ones that have been looked at in all the species of mice, none of them are identical to the viruses that are being said as circulating in humans. They're close, but not identical. So contamination's not very likely.
So even a lab mouse here in our building is going to have this virus in its genome. It's an
Absolutely.
endogenous virus.
Yep.
Do they produce infectious
Some do.
viruses?
So there are known. So this is the great thing about this. People were excited to find the virus because they were so familiar with this family of viruses. So all mice have endogenous proviruses in their genomes. They're of a variety of families. There are a lot of ways to group them, but one of the most useful ways is by the receptor that they use. So there are the so-called ecotropic family, which use one virus receptor, and the xenotropics, the one we're talking about here, which use a different receptor, which is known as XPR1. There are polytropic and others, but the xenotropic are well known, and they've been studied lightly since the 70s. They were actually initially picked up. These true mouse viruses were picked up because whenever someone passed a human cell line through a mouse, for example, you would do with a tumor in a nude mouse, or a human line passed through a mouse for any reason, just to amplify or study the line, it would acquire these viruses. The human cells have the receptor for the virus, the mouse does not. So the mouse would carry the endogenous virus, even sometimes have replication competent versions of the endogenous viruses, but the mouse would never amplify them or replicate them because they don't have the receptor. But if you put a human cell line into the mouse, it immediately picks up this virus and does replicate it. And when you take the tumor back out of the mouse, or the human line back out of the mouse, it would always almost acquire that virus. So people were very excited about that in the 70s. they thought it was a human virus that had been induced, but no, it was simply the mouse virus that had been picked up by the human line.
Do we know what the receptor is for Xmrv?
Yes, so it's this. We don't know much about it. It's a membrane surface protein called XPR1.
Okay.
Function largely unknown.
Seems similar to many of the mouse receptors
Yeah.
for mouse viruses.
Yeah, I mean they're quite diverse, really. The various murine leukemia virus receptors, but it's not an unusual surface molecule.
So they're called murine leukemia viruses. Do they cause leukemia?
Yes.
So the endogenous copies that are producing virus in many mice, that's causing leukemia?
So the ecotropics, yes, those that can actually replicate in mice will cause and do cause leukemia, and they can be that can occur in at least two ways. You can actually just infect the mice at birth with one of these viruses as an exogenous infection. The mice will become viremic, and they will get leukemia, and they will die. So the Maloney virus that we work with is just such a virus. If you give almost any strain of mouse at birth Maloney, it will replicate, and the mice will virtually all be dead in a few months of leukemia.
If you infect an adult mouse, what happens?
Well, they tend to get control of the infection by the immune system. They don't get as viremic.
Right.
They recognize the virus clearly as foreign, and they suppress it most often enough that they don't get leukemia, because leukemia requires the infection of a very large number of cells. And we now appreciate that long lag is because of the need for the virus to, by chance, insertionally activate some oncogene.
So could Xmrv infect a mouse?
No, not per se, because they don't have the receptor. There are rare mice that do, wild mice, and rats do. And we know that the difference in the critical amino acids of the receptor are the basis for that. But given the similarity to the ecotropic viruses, which do cause leukemia in mice, and everything we know about the xenotropics, even in the 70s, we were certainly aware of the possibility that they could cause leukemia in man. And we worked with the virus pretty casually, I have to say, back in the 70s. But the history was, even then, that people who'd been working with these viruses for years never showed any infections. And more only now, really, have people started putting them into primates. There now some work being done in companies are seeing infections in primates and following the viremia. Nobody's yet seen disease, but they have certainly seen virus replication in a variety of tissues out many months, years in primates. So, I think the potential that they could cause disease is out there.
So how has the prostate connection held up since 2006, I guess it was?
Yeah, so we're now seeing it in a small number of prostate samples. The range of prevalence depends on who you ask. It's from very low, I guess in some subsets of people, 0 in some labs' hands, to percentages as high as 20% and up in the case of our friend Ela Singh, now in Utah.
Are these all American studies? There's some European studies.
There are,
well, right? I think there's a German study.
Yep, and I would say the American patients are being seen to have higher prevalence probably than the European. Don't know if that's real, whether it's just experimental. One of the issues.
It is found in European patients, so it's not geographically restricted.
I can't remember if the European studies had any. I think yes. I think there were rare cases.
I have German studies, 0 out of 589.
Right.
0 out of 105. So there were 2 separate German studies, and there was one in Ireland too, 0 of 139.
The European ones are very low.
So you went to CROI recently,
right?
This meeting in San Francisco. Did you hear anything more about that?
Yeah, I mean there were some of those studies were reported.
Nothing positive though.
Well, there were several low levels in prostate cancer in America, certainly.
Right.
But the numbers are low.
Okay. So does it make sense that this could cause prostate cancer?
Well, it's possible. You wouldn't ordinarily think of the virus as being specific for any tissue. The leukemia viruses are leukemia viruses largely because they grow in cells that are rapidly dividing.
Right.
And we presume that's why the murine leukemia viruses cause leukemia as opposed to a sarcoma. But because otherwise insertional activation could happen in any tissue. So could that be the case here? Yes. And what would make it tissue specific? Well, it could have been, for example, the distribution of the receptor levels. We don't think that's the case. XPR1 is not high, particularly in prostate tissue. We've focused in my lab on the LTR, that is the promoter for transcription from the virus, and that is interestingly very hot in prostate cell lines. Mm-hmm. So certainly that could account for a tendency of the virus to replicate more vigorously in prostate tissue. We don't know how. We haven't done a complete survey. I don't think anybody has yet of all the tissues in which the promoter might be very hot, but it's certainly very hot in a handful of human prostate cancer cell lines. Surprisingly hot to us. It is as active in those cell lines as the Maloney mouse virus is in the best mouse cell lines. Mm-hmm. So it's a real virus, and it can replicate very vigorously in culture, more so than we ever thought. Yeah. I think nobody studied it much since the 70s because of the general feeling that it was an incredibly weak virus, that it was going to be difficult to study because it barely replicated in most cell lines, and that's true. But now, having found that it does replicate really, really well in some cell lines, the studies will be much easier.
So has anyone looked in different prostate tumors at the integration site?
Yeah, so that's being done still now as we speak. Nobody has found it and this is an important negative result, so nobody has yet found it clonally integrated next to an oncogene in a tumor. The early studies are still such that we don't even really know the cell types that it lives in. People are just doing sections and ELA and others like that, and are seeing it in more cells types than originally thought. There were initially concepts that it was only in stromal areas, not in the tumor itself. Seems to be in both now. But to date, nobody's seen the kinds of things you see with leukemia, which is an insertional clonal insertion next to an oncogene like MYC that was clearly a clone triggering a tumor. So we don't have any reason to feel confident that that'll be the case. So prostate cancer is interesting. It may be not consistent with that anyway, in that it's a disease where there's a long phase of sort of chronic, maybe immune stimulation or irritation that might be consistent with some other mechanism than insertional activation. Maybe the tumor is the result of inflammation or expansion of cell lines in a non-clonal way, may be triggered eventually into a clonal tumor, but the virus may not be the direct immediate cause the way it is in leukemia.
Like Hep B, where you don't have any oncogene activation, but there's a long-term inflammation that may be involved. So the one thing that I always wondered about was, you have a very high homology of this virus with mouse viruses. So can you learn from that how recently it went from mice into people?
You can, and there's some issues there for this particular virus. Normally that's right, and the similarity is so great that people who really think about this deeply, like John Coffin, would say this had to be relatively recent. This had to be a fairly recent transmission.
10 years,
or
20?
Oh, more. But I mean, you couldn't narrow it down that much. But thousands, not millions of years ago. Okay. Now the problem is that the various isolates that have all been sequenced from humans are unbelievably similar to each other. Very, very similar. Even more similar than the close similarity to the mouse
but not identical,
viruses,
right?
But not identical, but very similar. And that's very odd because viruses that we're used to, like HIV, are very prone to mutation, and they very quickly evolve and diversify. So that in the case of an HIV-infected individual, we think of the person as infected with a swarm of virus, not with a single virus, really,
Right.
because when you pull individual clones and sequence them, they're different. This is not the case, apparently, with Xmrv. And so there's something funny about its lack of evolution across a person or across different people.
So that would be consistent with it just being integrated in the genome and not
Right. So that would be one thought, is that it's infecting a person and then replication is slow or not happening. And therefore you just retain the original copy, basically, that you were infected with for a long, long period of time. But it's not clear if that's the answer to this slow evolution. HTLV1 is a little like this. HTLV1 also is not as diverse as many other viruses, and perhaps its replication is also restricted in some way in humans.
So this is a biological issue. It's not troubling in terms of the origin of the virus.
Not
It's
really.
something we need to understand.
It might affect things like how often is transmission happening,
Right.
and how many independent transmissions from mice, let's say, to people have happened. And in contrast, how often is person-to-person transmission going on? And we don't know anything about the routes of transmission, except by analogy to what we know about other viruses, where you would say, well, you need blood sharing or you need sex.
Could it be sexually transmitted?
Could be.
Have people looked in semen yet?
Yes, and people have also seen stimulation of infection by proteins found in semen.
That's right.
So there's
static fragments, right?
Exactly. That's not unique to Xmrv.
Yeah.
That's true of a lot of retroviruses, but consistent, again, with the possibility of sexual transmission.
So what do you think, what has to be done to show that this is the causative agent of prostate cancer?
Well, the smoking gun would be to find the virus integrated next to an oncogene in some subset of the tumors.
Right.
That may not happen. That would be compelling if we saw that. Otherwise, I think you just need better epidemiology. So one of the things that's really critical is that we need reliable, reproducible assays for the virus by PCR, probably. We need good assays for seroconversion. Because there are claims that people are seropositive against the virus.
Yeah.
We need those to be done widely to get a sense of what is the real rate of infection in the population at large, in people with prostate cancer or other diseases, because we don't really know the range of diseases in which it may be found. And that brings us, I suppose, eventually to the chronic fatigue issue.
If it were clear that this virus was involved in prostate cancer, it's not going to be causing all prostate cancers, right?
No way.
Just a subset.
Yeah. I mean, even initially, it was only seen in that subset that are familial due to this inherited gene.
But that's no longer the case.
And now it looks like it's being seen outside that subset. But the numbers remain small in terms of percentages of people.
All right. So then at some point, the people out in Nevada, what is the Institute, the Whitmore?
I can't remember the name.
WPI.
Yeah, okay.
They reported in Science that the same virus, essentially, but not exactly the same, but
very close.
Similar,
yeah.
They found in many chronic fatigue syndrome patients.
Right.
And now the whole field has exploded.
Right.
Because are there more people with CFS than prostate cancer in the world?
I don't know the numbers. I mean, the chronic fatigue diagnosis is a very difficult one.
To do, yeah.
It's almost by exclusion. I mean, there are now criteria that are used to score it, but it's first of all, one has to eliminate all the other causes of potential fatigue, and what you're left with is pretty much chronic fatigue syndrome. But it's been an issue for many decades, whether it even existed as a disease. I think it's well accepted now that it is. And then there was another long phase where many different viruses, bacteria even, were ascribed as the cause. EBV was a popular one for a while. And I think all of those one by one have been ruled out.
How do you rule them out? By just showing that they're not prevalent in most cases?
Yeah, there was no correlation that the viruses were in people with and without chronic fatigue equally, and therefore there was no link, essentially.
So the first study that was published in Science was a huge number positive of chronic fatigue patients. The first time that those kinds of numbers had been found, right?
Well, certainly the first time things looked as good in terms of correlating with a given virus, right? So this looked very striking and unbelievably exciting if you were in the field.
And they also recovered virus from some of the patients.
Right. Yes, Frank Rossetti said it's in plasma, and he was able to recover live replicating virus out of some of these people. So all the signs look good, and today still do look good as far as one can see in this paper. But it's certainly also true that now similar studies with even larger numbers of CF patients have been looked at in Europe and elsewhere, and they're not seeing it. So, either there's geographic issues, there may be strain issues, there might be PCR primers that people are using that don't work for some strains of virus. One base pair is all you need in the primer to cause you to miss that. So I think at this point, the CF connection is pretty much up in the air. I don't feel confident that I would vote one way or the other yet, and I just think we have to get more data.
Of course, the people who originally found it are very vehement that it's definitely the cause and that everyone else doesn't know how to do PCR.
Right. Well, I think that they're at least adamant that it's real, that the linkage is real. And I guess they also are pretty strongly coming out about cause. And I think that's a very tough call to make at this point. There's no way I would feel comfortable claiming that this was the cause of either prostate cancer, any other disease at the moment.
They claim to have data from all over the world implicating the virus in CFS, but we haven't seen that in the literature. So until we do, were they at the meeting, Croy?
Not much, no. They weren't. That crowd was not at Croy.
They should have been,
They should have been, probably. Well, there are more workshops happening all the time. There's one coming up in the next month at the NIH. So, I think all this will come out.
So you need more data.
Yeah.
That's what it comes down to.
Basically.
It could be geographically limited. It could be a detection problem.
Yep.
I have a review here by Joachim Denner, who says that in cases of low proviral load, antibody can indicate infection even when PCR doesn't. Is that true?
I'm not sure what example virus he's talking about.
HIV,
but
I think.
But I think certainly you could, because PCR can be done at any level of sensitivity, including bad, you could certainly imagine a virus having been cleared, HIV not being cleared, but non-retrovirus being completely cleared, so there truly is no signal that you could possibly detect by PCR, and the person would still retain seropositivity for decades. So certainly that is a possibility. And I think it's also very compelling if you do have seropositivity because, it's not contamination anymore as an issue. You've got a person who's got an antibody response. He probably had to be infected.
Sure. So this is also found in the original study in healthy people.
Yes.
So that's troubling, right?
Yeah. That raises a lot of issues. Is it in the blood supply, for example? We're certainly not at the state yet of monitoring blood donations. That might come to pass if it's ever found that this is really a pathogen and is really prevalent at a worrisome level in the population at large. But that's why we really need this epi data. We really need to know what is the prevalence of this virus.
So people are clearly doing that, right?
Absolutely. Yep.
So it seems to me that one of the ways you could resolve the difference between the U.S. and the European studies is to swap samples.
Right, and that typically, amazingly, has not yet been done very much. Very few.
We understand.
No, I don't know why. It's some of us looking at these papers are amazed that they didn't. Yeah. I guess people just are working very fast for one thing. They're trying to get stuff out quickly, and they haven't taken the time to swap samples. So these studies that have reported zero recovery out of hundreds of samples, of course, they did the positive controls of clones of DNA of the virus, and so they know their PCR is working at that level, but they haven't taken true samples from somebody else that that had a positive signal and shown that they could recover it. In some cases, it's happening, but the very earliest studies didn't take the time to do that.
So how might Xmrv cause a disease like chronic fatigue? What are some of the ideas?
Well, you could imagine various changes in the blood that would give you fatigue, I guess. You could imagine lymphopenias of various sorts. And some chronic fatigue people believe those are the kinds of things that have been seen already in the disease. So, the virus could be replicating in the blood and killing cells. Complement could be killing cells that are infected. The whole immune system could be working overtime, trying to reject the virus. There's a lot of reasons that you could imagine a retroviremia would cause fatigue.
So we don't still know what cells in people this virus infects other than blood lymphocytes, I guess, right?
And these prostate section tissues, which are either tumor or stromal cells. But so complete surveys haven't been done, and the best that is not yet reported, but that was talked about at CROI, were infections of primates, where they were finding it in a variety of tissues. So it's
what kinds of primates?
Rhesus, I think rhesus macaques.
Do they get ill?
Not yet. What they did, I think we're talking about half a dozen animals, not big numbers yet. The first set they followed over the course of about a year, and there was they saw a big spike of viremia, just as you.
How were they inoculated?
I bet IV.
IV.
Yeah, okay. I think.
All right. Spike of viremia.
Spike peaking at about a week or so, and then a big loss, but low level forever. I mean, for the rest of the year, and then they would at the end of 6 months or a year, they would take the animal apart and look, and they'd found virus in a lot of tissues. So it clearly is replicating. It's clearly there is a response to it that's pretty effective, but like any retrovirus, it never cleared. So then the second set of animals they took, they actually just infected them acutely and then killed them at the peak of viremia at a week, which they now knew was going to be at a week. And there they found the virus all over the place too. So it's a real virus. Yeah. It really does infect primates and probably most mammals that have the receptor. But does it cause any pathology? We don't know.
So did they observe, I guess they observed the animals for clinical lethargy. Did they do blood work, that, lymphocyte counts and enzymes, that sort of thing?
I think those were discussed, and again, we haven't seen the papers.
Yeah.
But my recollection was that, there was nothing apparent. There was no dramatic pathology.
So you could make a mouse model for this by putting the right receptor in.
Yeah, or you could actually, so we planned to do that at one point, so did a number of people, by putting the XPR1 into transgenic mice. But then we stopped, and I think a lot of people did when they realized that rats are naturally XPR positive, so you could do the experiment in rats.
I see. And, so do you plan to do that?
We don't, but others are doing that. Yep.
Have people looked at the integration site in chronic fatigue patients?
Not that I'm aware of, no. People have looked at the integration sites in cell lines that have been infected. People have done the kinds of studies that Rick Bushman has done in looking at integration sites across the genome for a variety of viruses.
Right.
And the result with Xmrv was that it's very much like MLV, which means that it tends to go into active genes, tends to go into the 5' ends of active genes, but it's not otherwise terribly restricted in any way. So it's clearly going into the whole genome.
Is the virus cytopathic in human cells?
No,
it's not at all.
Not that we've noticed. Now, Dusty Miller has looked and says he thinks he can see some morphological changes in some cells, very subtle. The xeno and the ecotropic are very benign, really, in most cells in culture. You can't look at a dish of 3T3 cells and look just looking at them, whether they're infected or not. There are reporter cell lines that are sensitive, however, and those can be used to make a plaque assay. The XC cell is one of them. There's another FG10. So there are lines that are sensitive to virus. They tend to cause syncytial formation. They fuse, and that's probably envelope mediated. They have some common features that probably make them useful reporters of infection, but they're rare, and your garden variety cell line doesn't have any changes whatsoever. Dusty Miller has also looked for transforming activity, because there are, although these viruses have no oncogenes in the classic sense, there are analogous viruses in sheep, the Yaxiki virus, where the envelope is an oncogene and is transforming. So he looked hard in a variety of cell lines for whether Xmrv could do that, could generate transforming foci. He couldn't really find any. The only example, when he pressed it, was he found an example where the virus had transduced the RAS gene from one cell line to another.
Yeah, okay.
So it's got all the capabilities of a traditional eco or xeno virus, but doesn't seem itself to be transforming.
So this virus, Xmrv, seems to be sensitive to AZT.
Correct.
But I don't think any of the other antiretrovirals, right?
I think there's a little paper surveying the RT drugs, and that's right. AZT is the one that's best. And so, it tracks with the behavior of all of the murine leukemia viruses in that regard.
A number of people with the disease have wondered whether they should take AZT, even now when still we don't know exactly, but is there any up or downside to doing that?
So the people I've talked to say they think that the side effects of AZT at this point outweigh the benefits, the potential benefits.
So you'll feel worse.
You'll feel you're more likely to have difficulties just taking the AZT, as of course many people do. It's not as though you couldn't live with it. People do. But
well, with HIV, which was fatal,
you took the side effects.
But here, although people with chronic fatigue would say that their lives are ruined without something, so you could almost equate it to being HIV fatality. So maybe they're willing to take the risk.
Right.
I get a lot of email about this and a lot of comments on.
I certainly wouldn't take AZT without knowing, if I had chronic fatigue, without knowing that I was viremic at least. If I was convinced firmly that I was actively replicating virus, I might contemplate it, and I was at the end of my rope in terms of the disease. I guess I might consider AZT as a treatment of,
as long as you understand that the side effects might be terrible.
Yeah.
Right.
Yeah.
But presumably other screens are going on now at companies, even in anticipation of finding this, causative agent of the disease. Yeah. Right.
Yeah. No, there's no doubt that the CDC is on the case. NIH is on the case. I mean, these data will come out, and we'll find out if there are pockets of infection or not, whether there are real focal infections locally, and we'll get ultimately a real sense of the prevalence, and we'll take it from there. But we don't have the data yet.
So should people stay away from rodents?
Well, that might be the case from a number of points, I guess, whether it's hantavirus or Xmrv or some other virus, probably.
But even lab mice produce these viruses, or similar viruses, right?
Yeah.
Which could be the origin of the infection years and years ago, right? It's a zoonosis, basically.
Sure.
Which may be now maintained in people.
Yep.
Do you think there are multiple reinfections from mice, or was it just one event?
Yeah, we don't know. I don't know. That would be an extremely interesting thing to know.
Are there other animals that have similar viruses?
Well, yeah. There's actually a very interesting case of koala bears in Australia right now. So this is an epidemic, true epidemic, and which is very serious. People have worried about losing koala bears.
Yeah.
Mostly due to habitat loss and others, but a very significant number, starting in the north of Australia and spreading down from the south, are getting infected with a virus that's exceedingly similar, actually, to Xmrv and to the mouse leukemia viruses again. In this case, they think they know the origin, which is a wild mouse on the northern coast of Australia. Those mice have essentially the virus that the koala bears have. They think it's a zoonosis to the koala bears. Yeah. And essentially 100% of them apparently now in the wild in the north are infected. About 50% or something in the mid coast and 0 in the south, but it's spreading south rapidly. And they're talking about isolating colonies to protect them from infection,
right,
before we lose them all.
Sure.
So it's so it does happen, and actually it's interesting, the fossil record, as it were, the DNA fossil record in all our genomes indicates that there are periodic epidemics of these viruses where a very large number of very closely related viruses enter the germline in a very evolutionary short time, and often in a geographically constrained way. So the mice in Japan will have acquired tens or hundreds of copies even of a very closely related family of viruses in their genomes, which were not in European mice, say. And then you can watch those migrations of mice by following that provirus.
Sure,
it's very cool.
Sure.
They're wonderful markers of events which are then preserved in the genome.
This virus, Xmrv, could go into the human genome.
Sure. And it has not yet, apparently. I mean, it's not
People
as far as we know. Yeah. And, people have talked about whether lentiviruses could do this too, could get into the germline. And
it hasn't happened yet, right?
Hasn't happened much as we know it. There are some hints that it may have things related to lentiviruses may be in the genome.
Human genome.
Yeah.
I think they're in the lemur genome.
Yeah. There are a few integrated copies.
Virus, yeah.
And there are certainly human genes that can complement some of the lentiviral genes, actually. Brian Cullen has shown.
So why would a certain percentage of people have the virus, presumably be infected and be well?
Yeah, I mean, who knows? The leukemia viruses in mice are very strain dependent. So if you have the right background, you can be infected and be fine. As a mouse. So there are clearly innate immunity functions. There are adaptive immunity functions that are sufficiently potent to suppress the virus, never clear it, but sufficiently potent to limit it to the point where you don't get disease. And I guess that's the case. That's even true with HIV in people. So
well, there's some very clear mutations that make people resistant, like
the receptor, the co-receptor. Yeah.
So in that sense, it's worth studying people with the virus that don't get ill to see if we can learn something that might be useful.
Sure. I mean, at this point, anybody who's demonstrably infected is going to be studied intensively anyway to see if they're well or not well.
So this is ongoing. So basically, within the next year, we're going to get a lot of information, right?
I would think in a year we should know much, much more.
This paper that I found, it's a review of the Xmrv with respect to xenotransplantation. And his point here is that pigs also have similar viruses, so we better watch out.
Right. So Jonathan Stroy and others raised this some time ago. So those are the PERVs, so-called, the pig endogenous. Yeah, I know, scary. Anyway, the pig endogenous retroviruses. And, when organ transplants were being considered on a large scale and pigs were potential donors, somebody thought to ask early on, well, yeah, gosh, what about the endogenous viruses in pigs? Are they going to be now relieved from any immune restriction and now freed to replicate and cause problems? And so people have looked hard and they haven't seen them popping out, but they're there. And I think it remains a possibility. And there are families of them. They have different receptors. There's at least 3, I think, PERV A, B, and C. Families that use different receptors. So, pigs are no different than any other mammals in this regard.
He says in this paper that there's one study of 200 people who got pig material, and there was no viral DNA in these people, but they had, some of them, I think 3 had antibodies against GAG of PERV, but not against envelope. And so he's wondering maybe those were actually infections with Xmrv that cross-reacted.
I suppose.
Who knows,
I don't know how closely cross-reactive those are. But the other possibility would be that a PERV genome that's defective for envelope was induced. And there are certainly functional PERV copies, but also many incomplete PERV copies. And so it wouldn't be at all surprising for a GAG only or a GAG-PAL only genome to be induced to produce particles that would be nicely immunogenic and yet not involve actual infection.
In one of the UK studies of Xmrv and CFS, they found neutralizing antibodies in 22 out of 157 controls. But they said they weren't specific for Xmrv. They were broadly reactive.
To many viruses.
And they said it's caused by some other virus. So would that be another retrovirus that came from a pig or another animal?
Yeah, I mean, it would almost surely have to be another retrovirus to be, I would think, to be cross-reactive. But yeah, it wouldn't surprise me a bit if we're not exposed to other viruses like this.
He also says that because you get recombination among retros, it's possible that if you gave someone a pig organ and they had PERVs, that they could recombine with Xmrv and who knows, make who knows what. So he said you should screen all transplant recipients for Xmrv. Sounds like we're all going to get screened for it anyway in the future.
I mean, that is one of the things that we know retroviruses are particularly good at. And the reason it's not totally crazy to think about is that when you infect a mouse with Maloney virus, our nice, well-defined cloned virus, and you do get leukemia in the mouse 6 months later, the virus that's doing the insertional activation is almost never Maloney itself. It's always a recombinant. So
with an endogenous
virus. So this does go on all the time. So and any time an animal is viremic, virus is spreading from cell to cell, and it's not only transmitting itself, but it's transmitting any other viral RNA that's in a donor cell, and then recombining with it because recombination is unbelievably frequent. So you typically expect that any infection and viremia from an exogenous virus quickly generates a swarm of recombinants that are a mix of the exogenous virus and your whole genome's worth of endogenous viruses.
So this happens with DNAs or RNAs, the recombination?
The recombination, we think, is during reverse transcription, and that means that it requires that there have been a particle assembled at some point that contains both the parental virus and the endogenous.
I see.
The RNA of retrovirus is always packaged as a dimer, and the recombination is certainly most high frequency when the two genomes are packaged together into one particle, and that particle then infects the new cell, and that's when the recombination happens.
Could that happen in humans right now? Because do we make RT?
We don't have high levels, at least, of anything known that would be a likely candidate to recombine with Xmrv. People wondered about any AIDS patient or HDLV1 infected patient, whether they would be generating recombinants with anything from endogenous genomes, but we haven't seen it.
Yeah. Even though we have a high percentage of endogenous retroviruses in our genome.
I mean, 8% of our genomes by weight are made up of some kind of retrotransposon or virus. Yeah.
You know, someone just did a screen of all the genes that have ever been sequenced, and the most common type of gene is a transposable element.
Right. I think that's right, that one of the most abundant single genes is reverse transcriptase in our genomes. Most of them, inactive and so old that they have become mutated out of function.
But that's because they can jump around, right?
Well, they can. I mean, even non-functional copies can be rescued by functional viruses.
Right.
So they can be amplified even if they're not functional anymore. But it's also the case that in these epidemics that we're talking about that may have happened 1 million years ago, there could have been introduced tens or hundreds of copies, and then they decay with time from that point on.
Amazing. So do you still work on Maloney in your lab?
Oh, yes, very much.
Because I know you do HIV and now Xmrv, still Maloney.
Yeah, we still do Maloney because it's the virus we know the most about, actually. It's very easy to grow. We have, wonderful reagents to do the study, much as you still do.
Polio. Yeah. It grows really well. So what do you do with Xmrv in your lab? What are you trying to figure out?
So, I mean, there are many issues. I mean, there are the issues that you've raised about the receptor, but and the pathogenesis in animals, and other people are doing that. But we've focused on two things. One is the promoter for transcription. We'd like to understand more about why it's so hot in prostate cells. Part of it we know is hormone responsiveness, and in that sense, it's like MMTV, mouse mammary tumor virus, which is a glucocorticoid responsive promoter. The Xmrv one doesn't seem to be that. It's maybe it's androgen responsive, but it works very well in some cells and not others that are otherwise equally androgen responsive. So there's something more to it. Mm-hmm. So we're just interested in knowing the positive elements in that promoter. And the other thing we're interested in is the interferon susceptibility or sensitivity of the virus. And that's been a long, complicated issue for retroviruses in general. People don't really understand when the virus triggers interferon production, and they don't really understand when it's interferon sensitive. So we're looking at that because we know this virus is at least interferon susceptible, interferon sensitive.
So there could be at least a temporary therapeutic approach, right?
To suppress it, yeah.
Even though interferon has got all kinds of side effects.
Right.
And people with Hep C get interferon and they feel
terrible.
Bad. Yeah. So it's not great. Do you get a lot of people wanting to come to your lab now and work on Xmrv?
A few, not yet too many. I think it's too new for people to commit their careers, their lives to yet. I think people are mostly taking, as would be appropriate in my view, a wait-and-see attitude for a year or so and see what the data really say about the prevalence.
Because it's not really a brand new virus. It's very much like Maloney leukemia virus, right?
Yep.
Which you've been studying and many others have been studying for decades, right?
Yeah. So I think, most of the things we know about Maloney will be true of Xmrv. It's really just I think of it as just a mouse virus with a unusual envelope, which means it has a unusual receptor that's not on most mouse cells, and yet is on a lot of other mammals. And I think we all would have been studying it much more intensively over the years if it had been as vigorous a virus as Maloney is in most cells, and had we known the right cell line to work with it, and we would have been certainly more eager to study it.
So the prostate cells are fantastic for Xmrv. Did you put Maloney into prostate cells too?
You can. So we compare the strengths of the promoter at least side by side with reporters, and this guy is as hot as Maloney, which is used as the standard of very hot promoters very often.
Well, the promoter is probably almost identical between Xmrv and Maloney.
No, not actually. The LTR, the U3 area is quite different. As it's of course one of the most rapidly evolving parts of any retrovirus is this area.
Yeah.
Even strains of Maloney vary among them fairly significantly. They get rearranged, duplications happen all the time, even in Maloney as we grow it. So this is different, and it has glucocorticoid response elements that are not in Maloney. So it's significantly different.
Neat, interesting story.
Yep. Okay.
Thanks for talking to me today. I appreciate it. And maybe if people have questions that they send me, I'll pass them on to you.
Sure, we can do this again.
And we'll do it again.
We'll have an update.
I have an update, absolutely. Since you're right next door, there's no excuse not to. I actually have also plans to get Ela Singh to talk with in a couple of months. She says they're doing some interesting stuff. Is she just focusing on the prostate angle, or is she moving also to chronic?
I don't know. I mean, all of the work she's done that I've seen has all been focused on the prostate, but I'm sure she's pretty interested in this whole CF story.
Yeah, I can imagine. I'm sure as a pathologist that she's looking into it. So we'll see. Stay tuned. Thanks a lot, Steve.
Great.
If you're a new listener to TWiV, please consider subscribing in iTunes. It's free and allows you to automatically receive each new episode as I post it. If you don't use iTunes, you can subscribe at the Zune Marketplace or by the RSS feed, or finally, you can download episodes at twiv.tv, where we also have show notes. If you like TWiV, please tell others about it. Links and reviews in iTunes are particularly helpful. TWiV is part of sciencepodcasters.org and promednetwork.com, websites where you can find high-quality science podcasts from a wide variety of fields. And don't forget to check out our other podcast, This Week in Parasitism, TWiP, at microbeworld.org/TWiP. I'd like to thank Dr. Stephen Goff for taking the time to speak with me. If you have any questions about his episode, send them to TWiV@twiv.tv, and I'll pass them on to him. I'm sure he'll be happy to answer them. You have been listening to This Week in Virology, the podcast all about viruses. Thanks for joining us. We'll be back next week. Another TWiV is viral.